Hello everyone, welcome to another video from Junior Basin. Today we are going to discuss about a process called agglutination. It is a type of antigen-antibody interactions. So the agglutination reaction occurs when an interaction between antibody and a particular antigen results in visible clumping. So the antibodies that produce such reactions are called agglutinin. And the agglutination reaction occurs when antigens and antibodies react in equivalent proportion. If the antibody is in excess amount, then it inhibits the agglutination reaction, and this inhibition is called as proson phenomenon. So, what is the agglutination process? Agglutination process or reaction is an interaction between antigen and antibody which results in the visible clumping, and the antibody which produce that reaction is called the agglutinins. And for that reaction, the antigens and antibodies they should have an equivalent proportion. Next is what are the types of agglutinations? The first one is the direct agglutination. In case of direct agglutination, no need of any particle or any inert material is required to detect the antigen. The example is hemolytic disease of newborn. It is an autoimmune disease. The first test is coombs test, direct test. The detection of incomplete antibodies on patient RBCs. Antibodies attached on the surface of RBC, then the patient RBC plus anti-human globin form the agglutination. And in case of indirect coombs test, the detection of antibodies in the patient serum, the rhesus positive RBC plus the patient serum, if contains incomplete circulating antibodies, coats the surface of RBC plus anti-human globulin which makes the breach and form the agglutination. We have already discussed about the erythroblastosis fetalis in type 2 hypersensitivity reaction. In that the maternal IgG antibody is specific for fetal blood group antigens. They cross the placenta and destroy the fetal red blood cells. The next type of agglutination is indirect, or you can say the passive agglutination. The passive agglutination it requires an inert particle such as the latex particle where the soluble antigens are coated, and the purpose of these inert particles they detect the antigen by the agglutination process. So the artificial carrier particle such as the latex built with antibody found bound to the surface, antigen present in a specimen being tested binds to the combining sites of the antibody exposed on the surface of the latex beads, forming the cross-linked aggregates of latex beads and antigens. And the size of the latex bead, generally larger size, enhances the ease to which the agglutination reaction is recognized. So you can see in this picture, this is the antibody and the coatings are the latex particles. So it forms the agglutination reaction. They form a cross link and aggregate to form an agglutination reaction. And the antibody it binds to specific site of antigen. The next is hemagglutination. Hemagglutination, as the name indicates, it is related to heme. That means RBC. So hemagglutination is used in the blood type. In typing the ABO antigens, RBCs are mixed on a slide with antisera to A or B blood group antigens. So we know that A, B and O. So during the process of blood typing, the RBCs are mixed on a slide with the antisera type A and type B, and the sampling checks to see whether or not the blood cells stick together or agglutinate. Agglutination means sticking. So, if blood cells stick together, it means the blood reacted with one of the antibodies. Determination of which antigens are present on donor and recipient blood is the basis of matching blood types for transfusions. So we know that for A blood group, the genotype is AA or AO, and the phenotype is A, and the antigens which are present is A, and the serum antibody is anti-B. In case of blood group B, the genotype may be BB or BO, and the phenotype is capital B, and the antigens are capital B, and the serum antibodies are anti-A. In case of AB blood group, the phenotype is AB. The antigens are both A antigen and B antigen is present, and there is no antibodies present. In the next O antigen, the phenotype is O. No antigens are present. Both the antibodies are present, anti-A and anti-B. So this is the hemagglutination reaction. This is the RBC. If some virus particle also attack, then it is called the viral hemagglutination actually. So they form the visible clumping. The structure of the A antigen, B antigen and O antigen. Let's first discuss the structure of O antigen. It consists of many polysaccharides with lipid long tail. So you can see it is a lipopolysaccharide. O antigen consists of glucose, galactose, N-acetylglucosamine, galactose, and fucose. If you add only the galactose with the O antigen, then it becomes the B antigen. And when you add the N-acetylgalactosamine to the O antigen, then it becomes the A antigen. In A antigen, N-acetylgalactosamine is the immunodominant sugar. And in case of B antigen, galactose is the immunodominant sugar. So that's all about the agglutination reaction. Let's summarize it once again. Agglutination means it is an interaction between antibody and antigens which form the visible clumping or sticky structure. So for that, the antigens and antibodies should have in the equal proportion. If the antibody is in excess amount there, it inhibits the agglutination reaction, and this inhibition is called the Poisson phenomenon. Then the first type is the direct agglutination where no inert particles or no coated particles required. And in the second, coated particles are required such as the latex beads. The latex beads generally enhance the agglutination reaction. And in case of blood grouping, hemagglutination processes occur. Just remember O antigen structure, then just add a galactose, then it will be B antigen. And if you add N-acetylgalactosamine with the O antigen, then it becomes the A antigen. So that's all about the agglutination process. Please subscribe to my channel to get more such videos. Thank you.